Hi, good afternoon, everyone. Thank you for attending the Lurie Cancer Center Core Technology Seminar Series. I'm excited to have Chad Haney here today to kick off our series for this academic year. Dr. Haney is the Managing Director for the Center for Advanced Molecular Imaging, who will be talking about how their core technology supports cancer research. So, welcome.
Thank you. Good afternoon, everyone. Hopefully everyone got a little bit to eat, and we'll go ahead and get started. Even though alphabet soup wasn't part of the menu, it's partly what I'm talking about today. So just a brief outline. I'll have a brief introduction, talk about the Molecular and Translational Imaging Core, preclinical imaging at CAMI, the Center for Advanced Molecular Imaging. And then the crux of this talk is what is MRI and what are all these acronyms which I've lumped together as alphabet soup. And I'll give an example for cell tracking, and there's examples for each one of those acronyms. And I'll talk a little bit about how we want to make a lot of this imaging quantitative. And with that, let's move into what is MTIC. The Molecular and Translational Imaging Core is a core in the Cancer Center. There's a group in Evanston, which I manage, and then there's another group on the Chicago campus. So on the Chicago campus, the Center for Translational Imaging is in Olson, and the Center for Advanced Molecular Imaging is in Silverman Hall in Evanston. And together we are the Molecular and Translational Imaging Core. So we offer magnetic resonance, optical imaging, X-ray, CT, which we just got last year. We have nuclear imaging in PET and SPECT. On the clinical side, we have several clinical scanners. There's three 3Ts and a 1.5T, and then maybe about 4 years ago, they added a clinical PET MRI. We have cell culture room infrastructure on the Evanston campus, and we have our own vivarium in the Center for Advanced Molecular Imaging, so there's direct CCM animal care there. We offer data analysis, both training users how to do it, and then of course we develop our own methods. And then on the Evanston campus we have this 25-foot 3D wall where we can display images. One of the things that's unique about CAMI is it was designed from the beginning to be multimodality. So you can see all of these modalities are within eyesight of each other. So if you wanted to do IVUS optical imaging and MRI, they're right across the hall from each other. So you're probably wondering, if we're in Evanston, how can you do animal work with us? Well, there's a van, not this van, there's a van that transfers animals between campuses for free on Tuesdays and Thursdays. If you need transportation on an off day, Monday or Friday, there is a fee for that, but the free transfers are Tuesday and Thursday. You can log into AOPS and do the transfer through the AOPS system. So this is a representation of all of the imaging that's available for small animals. The only one that isn't related for clinical use is optical imaging, but I'll touch on that very briefly. The only thing that we don't have is ultrasound. This is just to orient you in terms of scale. Down here is more of like molecular, DNA, things like that. On this end is whole body, and we're kind of in the middle for molecular imaging. And then to orient you with spatial resolution versus sensitivity, this is a guide that's for clinical imaging. You can see for resolution, CT is the best, and single photon emission computed tomography has the lowest resolution, and they're more or less inverted for their sensitivity to contrast agents. And then for preclinical imaging, you can more or less divide by 100 there to get the scale for clinical. So I'm just briefly going to talk about CT, and then the rest of the talk is about MRI. A lot of times you hear people mistakenly use those interchangeably, especially if they're talking about, for example, an athlete that has a sports injury. They may say, oh yeah, he went to go get a CT, when they really mean MRI and vice versa. So what is MRI and what is CT? So CT uses X-ray, so you're probably familiar with something like a chest X-ray. That's a 2D image. And the way that you make CT, which stands for computer tomography, or CAT scan, CAT is for computer-aided tomography, you take a 2D image, basically like a chest X-ray, at each angle while the camera moves around the object, or in this case, moves around the subject or the human. And the video that you see playing are all of the projections played in sequence. So it looks like the skull is rotating, but really the camera is rotating, taking a picture or a 2D X-ray at each position. And you use an algorithm to back project each one of those images to create a 3D object based on the attenuation of the X-ray. And that's all I'm going to say really about CT. One of the things that's different between small animal imaging and human imaging is the size of the object makes it harder to see soft tissue contrast in an animal. So this is a CT of a rat, and it's not uncommon in both clinical and preclinical imaging to have nuclear imaging superimposed on CT for anatomic guidance. And in a human, you would probably see the kidneys and the stomach much better. But in this rat image, the arrows point to the kidneys, and you can see an overlay of the SPECT image here. So this is a kidney, this is a kidney. And this collaborator was trying to develop an imaging agent to image the pancreas. And when he saw this, it's shaped similar to the pancreas. It's near the kidney, so it's in the right location, sort of. And so he got excited and he said, well, it looks like this is working. And I said, well, we really should do MRI because there's better soft tissue contrast. So that's MRI of the same rat, and that's the overlay. So now, if you're familiar with MRI, you can see this object here is actually the spleen. So his imaging agent was going to the spleen and not the pancreas. So this shows the strength of soft tissue contrast with MRI and the power of multimodality imaging. Anyone have an idea what this is? This is my litmus test to see if there's any MRI people in the audience. So this is called case-based data. This is what raw MRI data looks like. So this starting with this square here, as you go across, this is multiple echoes, and then this next one here is actually a second slice. So this is a 3-slice image with multi-echo. So how does MRI work? So it works by relaxation, 2 different types of relaxation of spins with protons. You can do other nuclei, which I have down there at the bottom, but we don't have time to get into that. So for this purpose, or for the purpose of this talk, we're mainly talking about protons. So I'm going to switch to this video here so you all can see that. So right now, these are water molecules spinning in a magnetic field, but the magnetic field is off. When you turn it on, the protons align with the main magnetic field. And so far, we haven't really done anything to get an image. The next step that's crucial to figuring out how to make an image, you need a radio frequency pulse. And you can see when you hit these water molecules with a radio frequency pulse, there's 2 of them that rotate, and then when the energy dissipates, they realign themselves with the main magnetic field. And it's that realignment that generates a signal that you turn into MRI. And now back to the talk. So the first example is called T1, that's spin lattice relaxation or longitudinal relaxation. And that is when the proton realigns with the main magnetic field, which is what I was showing. And then there's spin or a transverse relaxation, which is when you have more than one echo or more than one pulse, and then the dephasing is followed by a second pulse. And the difference between those is when you can get a T2 weighted image. T2 star is a little more complicated. I'm going to get to that in a minute. And like I said earlier, you can do other nuclei like fluorine and phosphorus and sodium, but we're not going to get into that. So here's the alphabet soup that I promised. Sorry it didn't come with your meal. So these are not an exhaustive list, but pretty close to an exhaustive list of the types of imaging of MRI and the acronyms that go with them. I'm only going to focus on these. So diffusion-weighted imaging is where you use a particular pulse sequence to see the diffusion of water, and if you get the direction of the diffusion of water, that's the second one, which is diffusion tensor imaging. So that's something that you may have heard of. Sometimes it's called tractography, and probably about 8 or 10 years ago, there was a famous DTI study with monkey brain, and it preceded the Connectome project where they imaged human brain, but this was a monkey brain, and they used DTI to show all the neurons and the nerve fibers and the directions that they go, and they're all color-coded based on the direction they go. It's a really interesting image. I think it was on the cover of Nature or Science. Anyway, now with the improvements in computing power, we can do that in a human. Back then, what they left out of the news with that monkey brain images, they imaged it over a weekend. So it was, I want to say, a 36 or 40-hour scan. So that's obviously not happening with a live animal or human. Moving on to the next one, dynamic contrast enhanced MRI, or DCE MRI. That's where you inject a gadolinium-based contrast agent, and you basically make a movie. So you image really fast, and at each time point, you're collecting data as the contrast agent moves throughout the body. And then you use math to do pharmacokinetics of the contrast agent, and that can tell you things about, for example, perfusion and permeability. And the last example is echoplanar spectroscopic imaging, or EPSI. It's very similar to bold MRI. So a lot of times when you hear fMRI, they really mean bold MRI. fMRI stands for functional MRI, and it's misleading because all these examples, whoops, let me go back. All of these examples here are functional MRI. So bold MRI is just a specific or particular version of fMRI, but they often get used interchangeably. But what bold stands for is blood oxygen level deoxy hemoglobin imaging. So you're imaging the change of oxygen from oxygenated to deoxygenated. This is an example of EPSI, which is a spectroscopic technique, so it's 4-dimensional. This is breast MRI that was done with EPSI at University of Chicago. So a conventional MRI, you have a single point on this echo. So if I go back, so it's a single point on here, all these echoes. Come on, here we go. And with EPSI, you're collecting all of those. And from there, you can get a spectrum for that single point. So for each voxel, so a pixel is 2-dimensional, a voxel is 3-dimensional. So for each voxel, I have a spectrum that makes it 4-dimensional. This is just a diagram of the pulse sequence. None of you are really interested in that. And then again, for each this is the K-space. For each position here, I have a spectrum. And what we can do is look at the shift from the center frequency, and that gives you information on the shift in water peak. And that's one example I'll show is it's known that the tumor environment is different than muscle, and so you get these shifts in the spectra, and you can turn those shifts into a separate image itself. So this is just different slices showing that. And then remember I said that for each voxel you have a spectrum. So this line is that particular voxel. That's what the spectrum looks like, and likewise here. So you can see that the tumor is quite different than the muscle. And you can turn that asymmetry into another image. And so this is called an asymmetry image. And you see how it's got contrast in the tumor. There's a dark spot where there's low signal, and then the muscle is more or less uniform. This kind of speckled appearance on the edges is artifacts from the edge. And again, I had on the outline that I try to focus on how we can make this quantitative. So you can see here the rim of the tumor has different asymmetry than normal muscle. So echoplanar imaging, it's fast, it's sensitive to motion artifacts, it's very demanding on the system. I'm going to go to a different example where we use a small particle iron oxide as a contrast agent. Can you use it clinically? Yes, that was a human breast that I showed. As far as doing this next step where you use a contrast agent to show the vasculature, you could do it in humans. I don't know if it's been done. I don't think it has been done. But let's move on. So this is a rat tumor. On the top is the tumor. On the bottom, it's a leg. So this is a tumor, and this is muscle. Again, tumor, muscle. And this is what the subtraction image looks like before and after giving the iron oxide contrast agent. And this dark region down here and here is highly vascularized. And we used a technique with a vascular gel. So this is a terminal procedure. You inject this gel into the vasculature and it makes a cast. And so this is CT now. So the bone and the cast look the same because you're basically filling the blood vessels with something that's radio opaque, just like the bone. So I should have color coded it differently. But anyways, the point of this slide is we superimposed the EPSI image, and this blue and red here are the vasculature that shows up in EPSI, and these are just 2D representations at the bottom to show that the vascular cast and the EPSI vasculature overlap. This is an example of arterial spin labeling. This is a mouse carotid artery, so the two arteries that go along your neck. And this is without contrast. You use a special pulse outside of the field of view down here, and then as the blood travels into the field of view, it's magnetized, and you do tricks with the pulse sequence to be able to visualize it. So this can be important in cancer research to show like antivascular drugs or changes in the vasculature as the tumor grows and starts to recruit, new blood vessels. This is a slide I borrowed from a colleague at Medical College of Wisconsin. It's not related to cancer, but it can be, especially in these brain models. So this is diffusion tensor MRI, and as I mentioned earlier, it's similar to diffusion weighted imaging, except it has directionality, and that's what's shown here. So this is a generic voxel showing the direction, showing arrows in all the directions, and then this is what it would look like as it's traveling down a neuron, and how you can kind of confine it to less directions. And then these are the different images that you get out of DTI. And then this is, this particular example, as I mentioned, it's not cancer related, it's a spinal cord injury model. And this is what it looks like. Sorry, I couldn't get the mouse going. So that's the injured axon, and that's the normal one. And that's what the DTI scan looks like of the spinal cord. Hopefully you can kind of make out the anatomy similar to the histology slide there. And then they added a different version where they do DTI plus spectroscopy and show that the change in the spectra can help you identify various lipids and quantitatively differentiate between mild and moderate and severe injury to the spinal cord. So with just DTI alone, it can be a little bit difficult to differentiate them, but if you add the spectroscopy, it better defines the different severities. Okay, moving on to DCE MRI. This is example from a 4.7 Tesla MRI. This is some older data. As I mentioned earlier, you use kinetic modeling to tell you things like perfusion and permeability. So this variable here is similar to perfusion, and this variable down here is similar to permeability. They're not directly measurements of permeability and perfusion because it depends on certain assumptions in the model. That's why I say it can mimic, or it can be a guide to tell you changes in perfusion or permeability, but not a direct measurement. This is an empirical model that was developed for smoothing. I'm going to skip over that. So this is what the data looks like, time along the bottom, and then gadolinium concentration on the y-axis. And so this is the baseline. You start injecting here, and you can see the gadolinium shoots up. And then the difference in shape, so the difference in this rise can tell you differences in perfusion, and then the difference in shape on this part, the washout part, can tell you information about permeability. So this is an example of a colorectal tumor xenograph on a mouse leg, and we're looking at vehicle versus sorafenib. Sorafenib is an older antivascular agent, and this was a study that I kind of inherited from a postdoc that got ill, and we were trying to make sense of the data and the way that it was collected. We weren't seeing any significance between the vehicle and the treated for both. So K-trans is similar to perfusion, and this V-sub-E is similar to permeability. You know, with your eye, you can see a big difference between day 4 vehicle and day 4 with the treatment. In this permeability measurement, they look the same. But statistically, we couldn't get anything, and we changed it to pixel by. So what the previous person was doing, he was looking at tumor versus muscle, so just the whole region of interest. And what we ended up doing is fitting each pixel to the model one by one, and from there we were able to tease out the difference that the difference that you can see with your eye. I mean, that looks pretty different from that. So it's just a little bit of math trickery that we were able to show that it's significantly different in this K-trans parameter and not this permeability parameter. So you probably hear a lot about artificial intelligence and machine learning, And one of the precursors to that is clustering. And this is an example of fuzzy C-means clustering. There's another type of clustering called K-means. But it's just a precursor to machine learning and then, to some extent, AI. So this is a project that I was working on. Again, it's not cancer related, but you can imagine this would work for tumors. In this case, we're imaging placentas. And again, the similarities between this and cancer is this is a model of preeclampsia where you have changes in vasculature to the placenta. So you can imagine with some therapies in cancer treatment, you'll get changes in vasculature and blood supply to the tumor. So there is definitely overlap. This happened to be newer data that I had used, this fuzzy C-means analysis, so that's why I'm presenting this instead of something that's cancer-related. I actually do have something newer that's cancer-related, but it's not published. And the co-author or the collaborator didn't give me permission to show it, so you'll have to wait till next year maybe. Anyway, oh, sorry, let me go back here. So this is an MRI of a pregnant rat. These are the part that are outlined are the placenta. These are the fetus. And then we volume rendered the placenta, and this is what the movie was. And then in addition to doing the volumetric study to show differences in size of placenta, we also did the contrast-enhanced MRI. So this is PBS treated, and this Suramin is a antivascular agent that mimics preeclampsia. And this blue and light blue is muscle tissue that's used as a reference. You can see it on this trace here and this one here. And these are color-coded to match these traces, and those are in the placenta. And the part that I'll draw your attention to, it's a little bit hard to see. On the normal placenta, you see this light green at the edge, so it's the interface of the placenta to the fetus. And on the preeclampsia model, you don't really see that. It's almost gone. So again, what we're using fuzzy C-means to characterize each curve, and then we can lump them together to get better signal-to-noise ratio. This is the so this is not parametric. So on the y-axis here is just the initial area under the curve. So that's not using any modeling with the kinetics. It's just looking at the curve and taking the initial area under the curve. And in this case, you see a difference between day 18 treated versus naive. And initially, the K-trans and the vis-a-vis, the perfusion and permeability didn't show any differences. But we did notice some differences in the muscle. And we looked at that a little bit more carefully. And what's interesting is the arrival time for the gadolinium is different in the muscles depending on which day or which part of the gestation period, which you can see day 19 versus day 17. That project ran out of funding, so we didn't finish it. So one of the moving back to something cancer related, I forgot to mention earlier that one of the things that we need to do, especially if we want quantitative imaging, is what is your ground truth? So in this case, we're looking at CD31 stain, histological stain to mark the blood vessels. On the left here is just a standard H&E stain. You can see the tumor quite clearly. This is muscle, and then this is the same tumor there. The point of this slide was to show that you can try different color maps to get different contrasts. So it's hard to see the CD31 label is actually light brown. I don't know if maybe you can see it a little bit better up here. Can you see the light brown? Anyway, if you change the color map, this red is the same as the brown that should be for the CD31. So to quantify this, you can do different tricks to help you with your quantification. So by changing this to a different color scale, you can easily pick out the blood vessels. Another issue with using histology and MRI is histology is destructive. This is an example of some code that we wrote to warp the histology to match the MRI. So this mesh here shows how much you have to distort the MRI to make it match the histology or vice versa. I have a few more examples. So this is another example with some clinical images. This is a human prostate. On the top here is a T2 weighted image. This is the histological slide. This is diffusion weighted imaging, and this is a T2 map. And we wanted to correlate them, and the problem is when you do I should say, this is ex vivo. So that's why the histology and the MR look quite similar. In vivo, what happens is this curve here in the prostate is from an endorectal coil. So it actually distorts the geometry of the prostate. And then when you do the ex vivo, you see how it's flat because it doesn't have something pressing against it. So how do we make the in vivo match the histology in the ex vivo? And that's where you have to, I don't think this this doesn't have the codec. It's fine. This is a 3D rendering of the ex vivo and in vivo. The in vivo, this shape here, you can see the endorectal coil is not only pushing it to curve it, but it's also pushing it to make it go down. And then on the ex vivo, it's flat and it's expanded. It's also a higher resolution. So this kind of stair-step appearance is because the in vivo is lower resolution. So this is 1.5 Tesla and this is 9.4 Tesla. So the difficulty in this project, there's poor sampling between in vivo and ex vivo, and the time between the in vivo MRI is usually quite a bit by the time we got the ex vivo sample. So we got the ex vivo sample right after the prostatectomy, but that's usually quite a bit after they get their final MRI. And then the time for biopsy is also on a different time scale. And then I mentioned the endorectal coil. I forgot to mention the bladder also pushes the prostate out of the way. And then there are problems with histologies. The formalin shrinks in non-uniform ways. And then, of course, there's tearing as you slice for histology. And also the slicing can be uneven. Fat is typically removed, and the fat shows up in the MRI but doesn't show up in the histology, so the volumes can appear to be different. But anyway, this is the example of when we got them to warp and match. This is the MRI here, histology, and these dots here are the tumor margins that the pathologist marked. And I think this one also, no, that one is also missing the codec. Okay, so I mentioned iron oxide contrast agent for EPSI earlier. This one doesn't use EPSI. This is for cell tracking. So this is a model of breast cancer metastases. So the cancer cells have been modified to have bioluminescence. So they have the luciferase gene, the firefly luciferase gene, but they're also labeled with the iron oxide particle. And this is a cardiac puncture model to simulate metastases. So we use the IVUS imaging to see if, basically to triage whether or not the animal got a good injection or not. And so in this case, the tumors are going to the brain as we expected, and this one is a bad injection, just stayed in the heart. And this is fluorescent imaging afterwards, ex vivo. This is a 3D rendering of an MRI, and the green dots are the cells. Same thing here, we just changed the transparency. And this is what the 2D slice of the MRI looks like. And these arrows point to the dark regions where the iron oxide is. And I believe this doesn't play, so we are going to play it here. So that's what the metastatic breast cancer looks like with iron oxide to track where they go. The kind of purple shell is the 3D rendering of the brain. And then it will show you the 2D slice that you would see, like not quite the raw data, but before you 3D render it. And then the last piece will show the vasculature using arterial spin labeling, which is similar to the technique I showed you earlier with the mouse carotid arteries. So the PI for this study left before we could finish. She's at Virginia Commonwealth now. But the idea was for the next phase to show that the cancer cells are going near the blood vessels. That's why they're superimposed that way. But unfortunately, she left before we could finish that project. Another version of cell tracking, this is with gadolinium nanodiamonds. You can see the cartoon here of how they are made. That's gadolinium. And then you load the cells, put them in the mouse. So one tumor has the gadolinium nanodiamonds, and then the other one doesn't. And that's what they look like in the MRI images. And then we color-coded them with a T1 map to make it quantitative. So I keep trying to drive home the message that where possible, we try to make these quantitative instead of just pretty pictures. And then this is over time the normal tumor, or the unlabeled tumor versus labeled and versus muscle. So the T1 map that's shown down here, we often do that in vitro first so that we can confirm that these molecular contrast agents do what we think they do. This is a slide I borrowed from a colleague in Colorado. We actually, I don't have it on here. We actually turned this into a white paper. So these are some of the things that you can, or these are the endpoints that you can image with molecular imaging to get quantitative imaging related to cancer endpoints. So for example, tumor volume, which I think is actually not a great indicator for response to therapy, because you can imagine you could kill a lot of cells in a tumor, but the volume doesn't change, or you can make a tumor swell, so it appears to be getting bigger, but it's not growing per se. So tumor volume is not the best endpoint, but it's common and it's easy to understand. Lesion number, that's using these techniques. Apparent diffusion coefficient is with DWI, which I talked about earlier. SUV is a standard uptake value from PET imaging, which I'm not talking about. K-trans I talked about with the DC MRI. That's associated with angiogenesis. And then these are also not MRI, so I'm going to keep going. I referenced machine learning and AI earlier. This is also adjacent to AI and machine learning. It's called texture analysis or radiomics. So everyone wants to have an omics, so the radiologists don't want to be left out, so there's radiomics. But radiomics is basically texture analysis, and the example on the slide here is you can see these two histograms are from these two regions of interest. They're two different types of tumors. And your eye can tell you this histogram looks different than this one. But then how do you get a statistic? How do you get a number to tell you that those two things are wildly different? And so you can look at things like skewness of the histogram, kurtosis. So skewness tells you how much it leans. Kurtosis tells you how much it's shifted. And then you can do things like entropy. And I don't remember what the NPP and UPP are. But anyways, this is kind of a precursor to machine learning, so you can feed all this into a neural network and get more information out of it and start doing some training and things like that. This is a collaboration with another core in the Cancer Center, well, it says CDT. It should be the Developmental Therapeutics Core. They're not a center anymore. But this is a iron oxide nanoparticle that was developed by Professor Dravid in Nuance. If you're familiar with Nuance, they do a lot of scanning electron microscopy and cryo-EM. So we did the MRI, DTC did the animal study, and then Professor Dravid did the chemistry. So this is the liver and blood vessel, and then the yellow is the spleen here. So these are the different profiles of a kinetic curve with this contrast agent. The last thing that I'm going to touch on is make sure you're doing the right statistics. I don't know if you're familiar with this XKCD cartoon. If you get a chance, look at the link below. The gist of it is they kept doing a student t-test, and they tried purple and red and blue, and then green was significant, so green jelly beans are linked to acne. So just a joke about using the wrong statistics, which are important when you're trying to do quantitative imaging. And with that, hopefully you learned a little bit about the MRI alphabet soup. Those are my collaborators that I'd like to thank. And thank you all for your attention. Do you have any questions? We have plenty of time. Or at least I think we do.
Yes, we do.
I do have a question. In the beginning of your presentation, you mentioned about with the MRI, you can evaluate the tissue edema and the blood vessel permeability. Could you explain that a little bit more? I was curious about that. The tissue edema and the blood vessel permeability.
You're talking about permeability?
Yeah.
Yeah, so for permeability of the vasculature, you need to use a contrast agent, and you do kinetic modeling of that to give you an idea if it changes in permeability.
What are contrast?
So there's gadolinium-based contrast agents. They're clinically available. So in those models, we use the same contrast agent that's used in the hospital. Yeah. So the key to that is you image really fast, and you do kinetic modeling of the curve, gadolinium versus time. Anybody else? Questions?
Anyone